Welcome to the presentation on DNA. This molecule is fundamental to the heredity and biological functions of all living organisms. Let's explore its function, structure, and significance. DNA is the hereditary material present in almost all living organisms. It encodes the genetic instructions necessary for growth, development, functioning, and reproduction. DNA is composed of long chain of nucleotides which store this genetic information. In eukaryotic cell, DNA is located in the cell nucleus and packed into structures called chromosomes. Each cell typically contains a complete set of chromosomes. For example, humans have 46 chromosomes. In prokaryotic cells, such as bacteria, DNA is found in the cytoplasm as a single circular chromosome and often contains additional small DNA molecules called plasmids. DNA is made up of building blocks called nucleotides. Each nucleotide consists of three components: a phosphate group, a deoxyribose sugar, and one of four nitrogenous bases, that is, adenine, thymine, cytosine, and guanine. These nucleotides are linked by covalent bonds between the sugar of one nucleotide and the phosphate of the next, forming a nucleotide chain. Next, we have the structure. The structure of DNA is a double helix where two strands of nucleotide coil around each other. These strands are antiparallel, meaning they run in the opposite directions. The backbone of each strand is formed by alternating phosphate and deoxyribose sugar molecules. In the center, nitrogenous bases pair through nitrogen bond. Adenine pairs with thymine and cytosine pairs with the guanine. So, in 1953, James Watson and Francis Crick proposed the double helix structure of DNA. Their model was based on X-ray diffraction images taken by Rosalind Franklin and Maurice Wilkins. This groundbreaking discovery explained how DNA replicates and carries genetic information. Watson and Crick, along with Wilkins, were awarded the Nobel Prize in Physiology or Medicine in 1962. Significance of base pairing. The specific pairing of bases adenine with thymine, cytosine with guanine ensures accurate replication of genetic material. Each strand of DNA can serve as a template for a new complementary strand. The sequence of these base pairs encodes the genetic information needed to build protein with each 3' sequence or codon according to the specific amino acid. Replication of DNA. DNA replication is a semi-conservative process, meaning each new DNA molecule consists of one original strand and one newly synthesized strand. During replication, DNA helicase unwind the double helix, forming replication forks. DNA polymerase adds complementary nucleotide to each template strand. This leading strand is synthesized continuously while the lagging strand is synthesized discontinuously, forming Okazaki fragments. DNA ligase then joins these fragments, completing the new DNA molecule. Key enzymes in replications. Several key enzymes facilitate DNA replication. DNA helicase unwinds the double helix by breaking hydrogen bonds between base pairs. DNA polymerase synthesizes new DNA strands by adding nucleotides to the growing chain and proofreads for error. Finally, DNA ligase seals the gap between Okazaki fragments on the lagging strands, ensuring a continuous DNA strand. So, what we conclude? The conclusion is that the DNA plays a fundamental role in storing and transmitting genetic information. Understanding its structure and function is crucial in genetic, molecular biology, and biotechnology. Advances in DNA research have led to significant scientific and medical breakthrough, highlighting its vital importance in the biological sciences. Thank you for your attention. Please do like, share, and subscribe our channel. Thank you.